So today I want to talk about SARS-CoV-2 evolutionary dynamics, but as kind of a nod to this being a promotion talk, I want to kind of cover a little bit of ground that's non-COVID. So in the last few years, since promotion to associate in January 2018, we've had a number of lab-led papers with first author from someone in the lab. And really the only thing I'd really like to highlight here is there's a lot of work in different viral pathogens. So we have MERS and dengue and flu and West Nile and Zika and Ebola and so forth. And so all of that's been going on. With the pandemic, there's a few threads that have been carried forward, but then a lot of effort has also been switched to SARS-CoV-2. So focusing purely on COVID, on SARS-CoV-2, and really what I'd like to highlight is kind of this very striking image here of all of the variant of concern viruses that people know so well at this point, alpha, beta, gamma, delta, et cetera. We're up to mu at this point. And this kind of evolution that's happened over the course of this last year has been absolutely remarkable and slightly terrifying. And I'd really like to kind of focus on this for the talk. So I want to cover 7 different things: this real-time tracking of SARS-CoV-2 evolution, emergence of variants of concern, current circulation patterns, assessing adaptive evolution, variant transmission dynamics, genomic surveillance, and some future perspectives. So real-time tracking. So since January 10th, 2020, continuously, we've been trying to keep nextstrain.org up to date, tracking this evolution of SARS-CoV-2. This happens through GISAID, where people from all over the world, groups from all over the world, are sequencing viruses, uploading to the GISAID database in close to real time as samples come in, and then we pull sets from this and keep this website updated. And this also provides kind of tooling for other groups to have their own views of different data sets. So we're now up to over 3.4 million SARS-CoV-2 genomes shared publicly. This is a absolutely astounding number, and nothing kind of completely unprecedented relative to previous pathogen genomic sequencing efforts. For this next strain of work, I'd really like to just briefly acknowledge Richard, Emma, James, Tom, Moira, Eli. It's been a huge effort from a number of people to keep this going. So if we go back to early 2020, up to March 15th, early on, the genomic sequencing was really most useful to track pandemic spread. So in that case, the mutations were basically just labels, where you're able to look at this labeled transmission chain and see how it's moving around the world. And so, for instance, we could track the spread from China into Europe and the US and so forth very early on. Early on, there wasn't a lot of kind of action in terms of adaptive evolution. So, or in terms of functional changes with those mutations. So the highlight here is going to be the spike D614G mutation that occurred. This occurs in China in January, subsequently spreads to Europe and US and so forth. And at this point, all circulating SARS-CoV-2 viruses are descendants of this single mutation event here. But early on, there wasn't a lot of things like this. So this was highlighted by being part of the spike protein. So spike is the critical for cell invasion of the virus. It's the primary target for a human humoral immune response. And it's really where we kind of, before watching evolution occur, where we'd expect a lot of the relevant evolution to occur. After this D614G change that happens in the very beginning of the pandemic, during, say, spring, summer 2020, we really didn't see much functional evolution. And in fact, due to kind of how severe mitigation was and how little international travel was going on, we basically saw all of these local lineages that are circulating in different parts of the world, and they're not moving from region to region due to lack of travel. In summer, fall 2020, we start to see the kind of first inklings of variant viruses, or of which are things like this spike protein 222V mutation and spike protein 477N mutation that spread locally within Europe, but didn't make it that far. And then we had in winter 2020, as everyone knows, the emergence of variants of concern, which was kind of a huge surprise for the community. I don't think anyone anticipated this level of evolution. So one of the strongest kind of characteristics here was the repeated emergence of mutations, of spike mutations 484K and 501Y across the world. This is the phylogeny up until April-ish 2021. These kind of turquoise viruses are the more ancestral forms. And you can see mutations to 501Y in alpha here and B117, both 501Y and 44K in beta here, or B.1.351, and similar parallel mutations in gamma in Brazil here. But you can see kind of those mutations occurring again and again across the tree. And there's just been kind of these three major clades that caught on. And if we look by late April 2021, we can see that Europe has been taken over by alpha, Southern Africa has been taken over by beta, and gamma is starting to really take over and has taken over Brazil. And this degree of spread in just a few months then, from emergence in fall to that degree of spread was really surprising and unprecedented as well. If we look in a little more detail here at the emergence of each of these three variant of concern viruses, so alpha appears to have emerged in the UK, Pango lineage B.117. In addition to 501Y, it has a number of other mutations in spike protein, including a very important 681H at the furin cleavage site, and mutations here in the near terminal domain that are thought to abrogate antibody binding. It has an entire gene, ORF8, that's knocked out, and it has this really kind of mysterious 3 base pair deletion and an NSP6 gene that it's not exactly clear what that's doing. However, we can see if we just look at time versus mutations away from the Wuhan-like virus, that this jump to B117 to alpha is quite distinct. And we have kind of the pre-variant viruses and then the variant viruses. There's extra mutations here. And then over the course of just a few months, we see that the older viruses are dying out and the newer viruses, the variant viruses are winning. We see a very similar picture if we look at beta or Pango lineage B.1.351 in South Africa. Here we have 484K and 501 spike, other mutations in the near terminal domain, and again, the exact same 3 base pair deletion in an SP6, and the same pattern at which the variant viruses are more mutated and then take over quickly in South Africa. And again, the same pattern with gamma or Pango P.1 in Brazil. We have again 484K, 501Y, a bunch of near terminal domain changes, and the same NSP6 deletion. And again, this looks more mutated, and then it takes over quite rapidly. So, so the working hypothesis here is that there's within host evolution occurring during infection, driven by natural selection against host immunity. So part of the reason this still maybe a bit speculative, but that we didn't see any of these appearing in spring 2020, summer 2020 during this first pandemic wave, but you only start to see these emergence of variant viruses in fall 2020 in second waves, especially in hard-hit areas like South Africa and Manaus and Brazil. And this suggests then this idea that the virus is bouncing from person to person, and during these second waves, occasionally encountered individuals who were recovered in the first wave, had only partial immunity, and then in those individuals, the virus and kind of its struggle to survive gets additional mutations that are aiming to either directly escape from that immunity, like 484K that abrogates antibody binding, or basically is able to survive in those infections by getting better at replication, like 501Y or 681H. So a bit speculative, perhaps some evidence here is that if you look at persistent infections in, say, immunocompromised individuals, this is a single individual who had COVID for 150 days. And during that time, the mutations arose that are some of these hallmark mutations seen in the variant of concern viruses. So we have 484K, 51Y, 144 deletion, et cetera. And so this is not necessarily saying that the variant of concern viruses always arise from immunocompromised infections, but just that prolonged infections within host evolution or within host evolutionary pressure tends to create the same mutations that are seen in the variant of concern viruses. So this gives an idea of perhaps why they're arising, or at least pressures that were pushing them to arise. Okay, so I want to move on to look at current circulation patterns. Oops. So if we look at over the course of this last year at how COVID's been spreading, we have the first emergence of these variant of concern, beta, alpha, gamma starting to spread. By spring 2020, 2021, these have kind of taken over the world. Delta starts to emerge in the late spring, summer. And at this point, basically everywhere is Delta, with the exception of South America. So here is looking basically up to today, a phylogenetic tree of all of these different circulating viruses. You can see this massive expansion of Delta here in kind of lime green. And at this point, if you look across the world, Delta is over 90% frequency of all SARS-CoV-2 viruses. It's taken over basically everywhere except for South America, which had its own endogenous variant viruses emerge. So in South America, it's kind of split between gamma in Brazil, lambda in Chile, Argentina, and mu in Ecuador, Colombia. And those have been doing well, but Delta is starting to invade in South America at this point. Interestingly, we see kind of South America also at the tail end of seasonal flu circulation patterns. This is just due to travel effects. And so it kind of makes sense perhaps that South America would be later. But I would expect going forward that Delta is very likely to sweep and become the common ancestor of all future SARS-CoV-2 viruses. Okay. So if we look across these variants, this is kind of the same sort of plot. This is the same sort of plot we were looking at before, where we have time on the x-axis, different viruses or each different dots here, divergences on the y. We can see at this point, most viruses circulating are about 40 mutations, 40 nucleotide changes, different from the emergence, the viruses from Wuhan in early 2020, late 2019. And you can see really interestingly that during this time when variant viruses are co-circulating with more basal viruses, that they have these additional mutations that then basal viruses start to die out and are more or less gone by the time we get to summer 2021. And so we have overall more mutations here. But if we look particularly in the S1 domain of spike, this is where most of the focus has seemed to be, or at least anecdotally, for evolution in these variant of concern viruses. So in this case, you can see that up until September 2020, there's like maybe 3-ish mutations that have occurred, maybe 4-ish in this S1 domain. But when we start to see these variant of concern viruses, they all generally have multiple mutations in S1 that kind of are showing then 6, 7, 8, 9, 10 mutations in S1 domain. And you can see this kind of fairly rapid increase then in the degree of divergence in S1 relative to the rest of the genome. Okay, so so this is kind of the slightly anecdotal pattern here, where it looks like S1 domain of spike is mutating rapidly, and that's kind of all that we can say here. And so what I wanted to focus on now is how do we actually assess adaptive evolution here and kind of do a bit more of a rigorous job to see what's going on with evolutionary pressures across the genome. Okay, so so this is going to be work that by Katie Kistler, assisted by John Huddleston in the lab, really kind of trying to dial in and assess adaptive evolution. This has just gone up to bioRxiv yesterday, so still pretty fresh. So here we took a phylogeny of 10,000 genomes where we tried to sample these as equitably as possible through space and time in order to avoid kind of database artifacts that if you just were to look at the database, you'll have many more viruses from the US, UK, et cetera. And so it's not using all of the data, but it's trying to kind of have a more, more subsampled, equitable view of evolution here. We can see, again, that we're highlighting some of these variant of concern viruses, alpha, beta, gamma, et cetera. Some other lineages that are considered important that are not necessarily variant of concern. We'll use these as colorings, as kind of guideposts throughout, but all of the analysis doesn't depend on the labeling. The analysis is actually looking directly at mutations and so forth. So what we do here then is the kind of primary thing we look at is clade growth as a metric for viral fitness or evolutionary success. So we can look across this phylogeny and look at each node in the tree, each common ancestor virus, and then see how many offspring, sequenced offspring does that node have. And if we look near the present, there's some that have more sequenced offspring more quickly and others that have fewer. And the ones that have more are growing in frequency. The ones that have fewer are decreasing in frequency. And so we can use this as a metric to kind of quantify that this virus, this node in the tree, is more fit because it had more, kind of by definition, it had more offspring than this other node that had fewer offspring. So it's less fit. And we can then use these clade growth estimates to then compare and see what causes a clade, what mutations cause a clade to be more fit, and what mutations cause a clade to be less fit. So, so if we look at S1, here we see the same picture that we were just looking at. Or if we look through time, S1 tends to accumulate amino acid changes quite rapidly. We go from non-variant viruses to these variant viruses that are colored here. So we have a rapid rate of evolution in S1, but kind of more critically, if we correlate mutations in S1 and to this clade growth rate, so effectively just the colorings here, we see quite a strong correlation. So this indicates that viruses that have more mutations in S1 tend to have higher fitness and are increasing in frequency compared to viruses that have fewer mutations in S1. And this, I believe, is quite a robust estimate of metric for adaptive evolution. It's similar to McDonald-Kreitman test, if people are familiar with this, but rather than just fixed differences versus polymorphisms, it's actually kind of a much more granular view of fitness via this clade success metric. So, so this gives us kind of a strong picture that we think that there's a lot of adaptive evolution in S1. As a control, though, we look at synonymous mutations in S1. And in this case, we see these accumulate over time. However, there's no correlation between synonymous changes and fitness and logistic growth rate. And if we do a similar picture in RDRP polymerase, where this is thought to be under, or assumed to be under strong functional constraint, we see a similar negative result, where viruses that are that have more mutations are not do not appear to have higher clade growth rates. So this is suggesting that our metric is kind of something that is at least giving false positives here. And then we can then apply this metric across the genome. And so we see different genes have different strengths of correlation, but S1 is systematically stronger than everything else. So we get some correlation, perhaps with S2 and spike protein, maybe some with N, M, and NSP6, maybe some with ORF7, but less than S1. And if we look across the genome, we see that S1 has the highest clade growth correlation. NSP6 and ORF7A also have significant correlations, but they have less, slower evolutionary rate compared to S1. ORF8 is a little bit of an outlier here. I mentioned that this was truncated or a knockout in alpha, and this actually happens again and again across the tree. So it seems that ORF8, this really high rate of evolution, is in part due to just a lack of functional constraint, and it's able to get knocked out fairly easily. And we have other kind of non-structural proteins and other more, more important proteins that are not associated with clade growth if they have mutations or negatively associate and that evolve more slowly. So S1 is really the highlight if we look across the gene, if we systematically look across the genome for evidence of adaptive evolution. And so if we kind of further dive in here, we can use this DNDS metric. So this is the non-synonymous mutations per site versus synonymous mutations per synonymous site. And this gives a metric of kind of how rapidly is natural selection enriching for non-synonymous changes relative to the supposedly neutral background of synonymous changes. And we see kind of the same picture that we had before, maybe a bit more anecdotally, that we don't have variant of concern viruses back in spring, summer 2020. There's not a lot of action in S1, but we start to really see things pick up in fall, winter, and then into spring and summer 2021. And at this point, DNDS for S1 is around 2. This is remarkable. Like usually on a full domain or protein level, it's rare to observe DNDS over 1. As an example, the HA1 domain of H3N2 influenza, which is this kind of prototypical, extremely rapidly evolving, adaptively evolving bit of viral protein, it's seasonal evolution rather than due to the pandemic, rather than in the course of pandemic. In the seasonal evolution, it has a DNDS of about 0.4. And so we're getting rates of adaptive evolution at the moment for SARS-CoV-2 that are about 5-fold higher than H3N2 influenza, which is I would not have expected this a year ago, which is completely surprising. We can watch this through time. I would anticipate as we kind of get a continued transition towards endemicity and we kind of the virus finds this low-hanging fruit for mutations that it can grab, that this will come back down. But I would imagine that it could end up landing much closer to H3N2 or perhaps even a bit above, rather than kind of more like seasonal coronaviruses or flu Bs, which are down here. And so this is suggesting then that there's a lot of potential for continued adaptive evolution in SARS-CoV-2. So we can also, with such a rich data set, we can dive in and look at very particular evolutionary dynamics here that are not usually available. One thing that was we wanted to look at was how bursty or how clustered evolution is at in S1. And we do this by taking paths through the tree where we go from the root to each tip, and along this path, look at S1, and we can see when S1 mutations are occurring. And we can get a distribution of wait times of how long in terms of months do you have to wait from one, or years here, from one mutation to another mutation to another mutation. And we compare this to synonymous sites and synonymous mutations S1 and RDRP polymerase. In synonymous mutations in RDRP, we don't see any enrichment compared to expectations of low wait times. Whereas in non-synonymous changes in S1, we see this peak here. So this is showing that there are bursts of like mutation occurring along these transmission chains for non-synonymous changes in particular in S1. And this is quantifying this kind of slightly more anecdotal observation that the variant of concern viruses tend to have multiple mutations. And it's showing that these occur kind of rapidly in tandem. This could be due to the virus transmitting through a partially immune population, or it could be due to some degree of epistasis, or both. We also kind of in an orthogonal way of looking at adaptive evolution, something that is really a hallmark is the degree of convergent evolution. If you see the same mutation occurring again and again and again, the same exact mutation, that suggests the work of natural selection. And so here we have a number of mutations that are occurring 4 or more times across this tree, which would be quite rare under neutrality. And so this helps to highlight very specific changes that are under natural selection, particularly things that when they occur again and again, each time they result in clades that have higher growth rate on average. And so that would highlight S95I as something occurring repeatedly and associated with high growth rates, 452R, which Delta has, 484K as well, occurring again and again and again. But also, really interestingly, this NSP6 deletion that I referred to before shows up basically as strong as anything in terms of its evolutionary impacts. And so something that, that definitely bears further functional characterization there. So, so generally here, the conclusion would be that mutations in S1 are rising via within host pressures, as discussed previously. This results in increase in viral fitness and replication, and then these become enriched in the viral population by natural selection. And then, although at this point, selection has been not primarily been for antigenic drift, it's really been to increase transmission rates as we kind of have everyone in the world have immunity to some degree to SARS-CoV-2, we can expect a switch towards evolution really being driven for immune escape and antigenic drift. And this degree of evolvability we've seen so far, to me, suggests that that we can expect a fairly rapid pace of antigenic drift in the future. Okay, shifting gears a bit, looking at variant transmission dynamics. This is really taking this work here that's looking kind of bigger picture and looking at specific variants and trying to look at their fitness and how they compete with one another. So here there's been multiple different approaches to modeling fitness differences between circulating variants. They each have their own advantages and disadvantages. I really like these multinomial logistic regression models. A number of groups have used these. So this is looking at 20 different states with data in the US. The dots are showing actual frequencies through time, so 0 to 100% of B117 alpha, P1 gamma, 526 from New York iota, and B.1.617.2 delta. And you can see that these multinomial logistic regression models actually fit the data really well. We get kind of a increase in fitness, but not quite as much, causing these alpha waves here, and then delta being more fit will kind of increase in each time. So that's those models fit quite well. However, they don't really capture a situation that we might often care about, where you could have something where there's a new variant moving through the population. It's fitter than other things, so its frequency is increasing, but still due to immunity and mitigation and all of the things that are happening, the overall case counts might be falling. And so I think it's helpful and important to have a model that would actually look at how case counts are behaving through time. This basic idea is trying to do that. What it does is it takes case counts that are reported by each state due to this kind of detailed case reporting system that we have now, and then it splits them based upon our estimated frequencies of each variant. And this helps to show that, let's say if we go back here, that in California, B117 or alpha picks up a lot, but that actually didn't result in much of a wave in California, but it did in Colorado, Illinois, Michigan, Minnesota, et cetera. So we have some states that have spring alpha waves, others that don't, New York with its own variant, et cetera, but basically everywhere starts to sees a sizable wave due to Delta virus that we're in the middle of right now, or perhaps just passing the peak. So here, Marlon Figgins in the lab has really dove in and tried to make a very rigorous approach to looking at these transmission dynamics. So, so Marlon has a Bayesian model here that has kind of a process model and an observation model, and everything is properly done. I won't go into any of the equations there, but I'll try to kind of give an overview of how the model works. So this is data from Washington State. And gray lines are case counts through time from January to today, where we can see now cases are higher than ever. And we also have a breakdown of frequencies that we actually kind of observe of if we look at sequences through time, how frequent is each different virus. And we can, again, in a similar idea, use this to estimate actual cases of each different variant and how many cases there were through time in kind of real numbers. And then from cases through time, you can estimate RT, or on average, how many offspring does one infection, one case leave going forwards. And from that, we can really kind of get a feel for what's going on. We can see that in the spring, RT is greater than 1 for most of the variant viruses, and they're increasing in frequency, but then dips in May, so they start to die out, and then kind of stays below 1. So all of the non-Delta variants are dying out. Whereas when Delta arrives in April, it has a much higher RT than other circulating variants, and it remains high up until mid-August. This data set cuts off August 15th. And we can see that here, that the growth advantage estimated for Delta in Washington State is significantly larger than other things that had appeared previously. Really interestingly, we can use this to look across states and be able to compare patterns. So each of these lines is showing a single state as a trajectory, the RT value and the RT value estimates over time for that state. So we can see that starting in March, non-variant viruses all have RT less than 1. They are dying out. The vaccination, natural immunity, mitigation, all the things that we're doing are winning. And like the pandemic would have been over if the variant viruses hadn't over for the United States if the variant viruses hadn't emerged. Whereas if we look in the variant viruses, we can see across states that alpha is over 1 up until May-ish, where it's dropping. Gamma as same pattern here, whereas delta is consistently above 1. So if we look then at this August 15th time point, we see all states are less than 1 for all variants except for Delta, which is consistently above 1 on August 15th. And we can use this to estimate variant-specific transmission rate differences. Each of these dots is showing the mean effect estimated for each state across the 46 states with enough data. And the main takeaway here is how consistent these estimates are, despite being kind of independent analyses across states. And this really suggests that there's this specific variant intrinsic effect for each variant in terms of fitness differences. And that can be quantified by looking at data in this fashion. So that's kind of the basic machinery. We're hoping to be able to take this in interesting directions. So this is the first stab here, where if we look across states and time points, we get an RT value, and we can make a linear mixed effects model to be able to try to see what vaccination coverage is doing to RT. And we can see that vaccination decreases RT across the board, as you'd expect, and that we decrease RT via vaccination across variants to a similar degree. I'd hope that this will start becoming useful if we get more drifted variants appearing to be able to quantify this hit on vaccine effectiveness in a kind of orthogonal fashion to how we might do things like neutralization assays and so forth. Okay, so generally, I'm hoping that in the future, we're able to kind of tie these two sections together a bit and really have granular empirical estimates of viral fitness through this really detailed frequency, enormous amounts of frequency data that we have, and that we can use these fitness estimates there and tie those together with predictors of which mutations or neutralization and so drop in neutralization titer, et cetera, to really have a predictive model of what actual genotype phenotype is doing to viral fitness. Okay, I'm going to shift gears again now. And talk briefly about genomic surveillance. So here, if we look across the world, this sequence sharing and this kind of endeavor to sequence SARS-CoV-2 has been amazing and unprecedented. We're up to, if we just look at sequences shared in August 2021, we have over 600,000 genomes shared in just August 2021. This is all data from gissaid.org, which is the primary database for the sharing and has really been fantastic to do data curation, et cetera, and act as a kind of a clearinghouse for this data. If you look at how this kind of behaves temporally, generally we have specimens collected through time, these dots here in blue, and the data is then sequences submitted. It takes some time to sequence it, et cetera. What I'm highlighting is that there's always something of a gap. You don't get a sequence immediately, but generally there's a leading edge in which sequences are turned around very quickly, and then some things that take longer and that kind of were collected a year previously and are finally sequenced. But for the most part, the turnaround times are quite rapid. And so then the metric that I like to use to think about this is, at this moment, if we look in the previous 30 days, how many genomes do we have that were collected and sequenced and shared in the last 30 days? And this helps us to kind of know what our situational awareness is. And we can see that across the globe has been continually improving. Again, remarkable. If we look on the last month, we have perhaps 200,000 genomes available in the previous 30 days at any given time. Again, remarkable compared to anything else and compared to a year ago as well. However, this genomic surveillance effort is certainly not equivalent across the globe. So the US, the UK, and Europe all have now stunning genomic surveillance programs. A lot of money has been invested here. The US has spent $2 billion to get this to occur. And US, UK, and Europe are all doing pretty equivalently in terms of just actual viruses sequenced. Other parts of the world are catching up and are doing a great job, especially compared to what things were looked like before. At this point, if we look in the last 60 days, in South America, we have perhaps 7,000 genomes. In Africa, we have perhaps 3,500 genomes. This is remarkable, generally compared to pathogen surveillance anywhere before pandemic, is probably enough to catch most variants. The biggest issue here is now not this count, but much more geographic gaps. So South Africa is really well sequenced, but there are other countries in Africa that are not sequenced as well. And so really kind of to get this global variant awareness happening better, it's mainly at this point not increasing counts, but trying to kind of fill in areas of the globe that are not getting sequenced at the same rate as other areas. However, the really detailed surveillance done by US, UK, Europe, I don't think we need any more viruses sequenced to find variants in these geographies. Where this becomes useful and where kind of these counts start to be a useful thing is that you can then start to compare this, if you have the data systems in place, to things like variant-specific hospitalization, variant-specific secondary attack rate, et cetera, and really understand the different epidemiology of the different variants rather than just kind of trying to find a new variant. And those are very, very different goals. Okay, so in the last couple minutes, I want to talk about future perspectives. This is going to be necessarily depressing. I apologize. So, so my broad expectations for what's going to be happening are based on a comparison with influenza. This is in large degree due to kind of my background where I have a lot of familiarity with flu, but also I think it is an apt comparison given what we've seen so far. So if we look at kind of these three key parameters, so the R0 of Delta, the number of secondary infections in a completely naive population, started out around 3 for SARS-CoV-2, but with Delta, it's now perhaps 5 or perhaps even 6. This is compared to an R0 of flu of around 2. And so, so that even with the same exact rates of evolution and immune waning in flu and in SARS-CoV-2, we'd expect much more circulation in SARS-CoV-2 just because it's more transmissible. However, we've seen that currently rates of adaptive sequence evolution in SARS-CoV-2 have been about 5 times the rate of H3N2 flu. I would definitely expect this to slow down, but this does suggest that the virus will be generally quite adaptable, and I'd imagine sustained antigenic drift because of this. Then if we think of infection fatality ratio, COVID had been about 10 times that of flu a year ago before before vaccination in these first infections. But with vaccination and this durable immune memory, you have consistently preserving 90% vaccine efficacy. This suggests that we'll drop IFR about tenfold for COVID. It could decrease further as there's like tertiary infections and so forth. But I'd suspect IFR of COVID around flu-like is a good ballpark estimate. So together, all of this suggests an endemicity in which COVID circulates every year. We have a COVID season. Due to R naught, we have a pretty high attack rate of perhaps 20, 30%, but that IFR is something more like influenza. So we get perhaps a something resembling a severe flu season with perhaps 50 or 100,000 deaths every year. So not at all like heart disease or cancer, and not at all like the pandemic, but a sustained public health burden that needs to be addressed year after year. So very briefly, some things that we can do to try to address this. I'm imagining antigenic drift will be a thing. We will see for certain, but we can try to get set up to have forecasting of evolution, know in June what we should put in the new mRNA vaccine for use in September in particular geographies, and actually have something that is really tailored to local circulation. We can be working on universal vaccines that either engender a broader immune response or target more conserved epitopes to have a vaccine that's more robust against evolution, and that we can keep this testing, contact tracing, public health apparatus going, adding in rapid antigen tests, and just try to have case-based interventions as a thing that we don't have to lock down society and so forth, but we can at least try to isolate cases and prevent onward transmission in a targeted fashion. In addition to improving treatments, improving ventilation across the board, there's a lot of kind of easy wins available as well if we just kind of invest in them. And with that, I will close. Really like to thank data producers from all over the world. The sequence sharing is kind of a remarkable human endeavor here. GISAID for their database and curation work, NextTrain team for tireless efforts, and really want to thank Katie, John, and Marlon for their particular work that I just spoke about. And with that, I will stop and take questions. Thank you.